All right, should we get started? There we go. There's the recording. All right. Well, we can go ahead and get started. I think the panelists are all here. I want to welcome everybody to the second Meet the Experts series. Today we have Steve and Kiran here to talk about their efforts preparing their IND applications, which I think will be really crucial to help our group understand. But also, I think it's going to be uh, a great session, and I hope the attendees have good questions for discussion. Uh, Ben is going to be our moderator for the session. All right, I'm going to turn it over to Ben and the speakers then. Thanks, everyone.
Yeah, I think the way we wanted to organize this was I'm going to start off with about a dozen slides and just kind of give an overview of the IND and the different components and some initial kind of tips or experiences. And then we're going to shift to Ben moderating some questions that people had sent in ahead of time. And then at the end, we'll open it up to, just a open forum for any questions or discussion from the group. So let's see. All right, looks like that shared. All right, and just a little bit of a disclaimer, I'm not a regulatory scientist, but I have been involved with a few different gene therapy INDs that have been submitted as investigator-sponsored trials. And so this is really more of me just sharing kind of practical experience and practical learnings of things that have worked for us. All right, so this is just going to be an overview starting off of the different IND components. There's basically 5 modules that are involved, and I'm going to go through these. So, there's module 1, which is basically mostly just administrative forms. There's something called an investigator brochure, which I like to call like the Cliff Notes version of the IND. And this is something that would be given to any of the clinical investigators or sites that are involved with the trial. And then module 2 is really where most of the content in terms of the work that our groups have to focus on. And this is just the summaries of all of your preclinical studies and the proposed clinical trial plans. Module 3 is all around the manufacturing, this, CMC, basically all the descriptions of your drug, how it was manufactured. And then module 4, everything that goes into module 2, that's going to be like a summary of all your preclinical studies. But module 4 is going to be a place where all the sort of formal reports of those studies are held as independent detailed documents. And I'll go into each of these in a little bit more detail. And then module 5 will be focused on the clinical trial protocol. All right, so module 1, like I said, this is mostly administrative forms, things like the drug label, anything about intellectual property, different forms that the FDA requires you to fill out when you're submitting an IND. You'll also provide feedback to all the FDA's points from the pre-IND and the Interact meeting. So this will be a place like all the feedback that you get from the FDA, all of that, or each of those points will require a response. And then the investigator's brochure, again, this is, like I said, this is a succinct document. I say succinct, but I mean, this could easily be, 20 pages or more or less. It should be a succinct document that really provides all the information that any investigators need. So kind of the background on the drug, what exactly it is, what the preclinical studies were done to inform its use, and anything that they need to know in terms of what the patients are going to go through, what the risks are. And then you'll have just an executive summary for the whole IND, kind of, what you're planning to do, why it's important. All right. So again, module 2, this is the component that I think most of our questions are going to be around. This is the component that I think the investigators here are going to spend the most time with. So you'll start off with kind of, background information on the disease, drug development history. You'll have tabulated and written summaries of all the preclinical studies supporting the safety and efficacy of the drug. So, these are really, and I'll go into this a little bit more detail. But so it's basically you have all of your preclinical studies are going to, or most of them should have kind of a formal report or write-up. That goes into module 4. But then in module 2, you're going to reference those full reports and basically just summarize them here. And same thing, you're going to have a tabulated written summary of the proposed clinical trial protocol, and that's going to reference the full protocol that's in module 5. So think about, module 2 is sort of the meat of your story. This is where the FDA is going to go, and a lot of their review is going to start here, and they're going to read through and try to understand the whole picture of what you're doing, and then they're going to go to the other modules to get the detailed information where they have questions. So your objectives and kind of putting them together module 2 and even thinking about how you're arranging your preclinical studies now is think about it like you're telling a story. You have to make it logical. You're going to walk the reader through it. So think about how you write a manuscript. And it's really the same principles here. You're going to start off with what disease you're treating, what's the unmet need that your drug will address, explain what your drug is, what its mechanism of action is, where does it go, how quickly does it act, how long does it stay around, go into some of the history and science behind the drug. So, just again, this is like you're writing a manuscript. You're giving the reviewers all the background information that they might need to kind of fully understand what your drug is and how it works. And then, your preclinical studies are kind of going to be divided into the studies that predict that your drug will be safe or effective to treat the condition, and then your studies that predict it will be safe, how you calculate your clinical dose, and then it's also going to have a component kind of focused on the clinical trial. So how are you planning to test the drug in human subjects? How are you doing this in a way that will treat them ethically and meet the clinical trial objectives? So again, it's really important to kind of set this up, give the FDA the background that they need to understand what you're doing, and then just sort of walk them through, like, this is the rationale for why we did this. Here's what we did. Here's the rationale that we, for, these are anticipated concerns. This is how we mitigated those through our preclinical studies. Here's the publications and data that make us feel like this drug should be safe and effective to test in humans. Okay, and then module 3, CMC. This is the one that everybody gets really bored with, that they don't want to talk about, but this is going to really be just chock full of a lot of detailed information about all the components that went into manufacturing your drug, how it was manufactured, how the analytical release testing was done. And ultimately, through all of that manufacturing, your goal is to say, look, this is manufactured in a way that it's going to be consistent from batch to batch, and there's it's sufficiently potent, and there's not any kind of impurities or things that would represent a significant risk to your patients. And I'd say most of this information should be provided by drug manufacturer, but I would strongly, strongly, strongly recommend that you engage an experienced CMC regulatory consultant and just make sure, because they're going to have to take all the information from the manufacturer and sort of format it and summarize it into the IND. And there's lots and lots of different subsections to this module. And so it's just making sure that all the information gets put in the right places, and the right level of information is provided. All right. And then module 4, again, module 2 is kind of a summary of all your preclinical studies. Module 4 is where, it's just going to be, it's almost like an appendix. You're just putting in independent study reports So these can be your toxicology study reports if you contracted a CRO to do your sort of pivotal GLP toxicology study. In my experience, those reports are like 500 to 1,000 pages, and it's just an independent report that you just attach in here, and then you summarize it in module 2. And then you have any of your other non-clinical study reports, and again, this is all non-clinical. So any in vitro proof of concept studies, study reports on off-target editing, your dose finding efficacy studies, anything about the pharmacology of your drug, where does it go? How long does it last? And so it's really, and I'll kind of go in, I'll touch briefly on these preclinical study reports. Like if these are coming out of your lab, what needs to be in them? And I think one of the pre-questions was asking about, hey, if you've got a manuscript that's published, then you might be able to just attach that manuscript and cite that manuscript, but you might need to supplement that with kind of some additional tables of raw data or something like that that just gives more information than what you have in the manuscript. So I'm going to say if you're doing studies in your lab, which we all are, and that the data from those studies needs to go in the IND, then if it's a really short study or simple study, you could just describe that and provide all the data in module 2. But the best way to do this would be for each of those studies to put together a independent study report. And so I'll say these are kind of like a manuscript. We can think about it like that, except you're going to have really an abbreviated introduction, abbreviated conclusion, and minimal discussion. So you're really just focused on whenever you're preparing these, you don't really want to talk about a lot of extraneous information. It's like the more information that you give to the FDA that isn't directly relevant, then it can just raise additional questions and maybe complicate their review, or it dilutes out the things that they need to focus on. So so you really just want to focus on, okay, why is this study important for the IND? What did you test? What did you do? What are the results? So you kind of have like the rationale for, why that study was done, what the results are, and then don't talk about the broader implications and everything else. Just really focus on why is this important considering the safety and efficacy of the drug for human use. And you're versus a manuscript, I think you're going to be kind of heavier on the details of the actual study, especially providing detailed information on the drug or any critical materials that you're testing. So again, if you have like a AAV vector that you're using, where was it made? What's the lot number? What are the characteristics? Do you have a certificate of analysis or something that you can attach? And so it's going to be kind of like a manuscript format where you've got your introduction, and then you've got your experimental details, and you've got your results, and your results are going to be kind of summary tables or figures. But then the FDA is going to want all the raw data. So what we typically do is we'll have a report that we write up, and then we just have a lot of appendices of tables of raw data. And so, the main report might only be 5 or 10 pages, but you could easily have like 20 pages or more of just tables of raw data. And one thing that I think is very useful, and I'd say best practice, is, if these are coming out of your lab, they're almost certainly not going to be good laboratory practice, but I'd say when we generate these, we always have sort of the key people that are involved in the study, that were involved with writing up the report. They would have a signature line. So they're going to sign off on the report, and then me as PI, I'll sign off on the report. So you kind of have a accountability trail. And then let's see, module 5 clinical trial protocol itself. So this is something that your group should, I think, just be developing, ideally even now, and moving along. And it's really going to be a very detailed document of what the clinical trial design looks like, every activity, every medication, every assessment that involves the patient throughout the entire clinical trial should be justified and described. You're going to have stopping rules, oversight, how the oversight of the clinical trial it happens, the statistical analysis plan, different, I'm not going, this isn't all inclusive, but you'll have the informed consent in here, and, all the known and hypothetical risks should be described, and how, what your strategy is to mitigate them. All right. So I've got a few, I think 3 slides of like general tips. This is a big elephant. How do you eat an elephant? One bite at a time. So don't get overwhelmed. You start off with kind of an outline or list of all the planned reports and sections, and just work through them piece by piece. And if the contributing preclinical investigators, basically the lab or labs that are doing the bulk of the work, I'd say focus on the study reports first. And then, once you have those study reports done, then a lot of times you can just take the conclusion paragraph or section and just paste those into module 2, and maybe add, just a little bit more information to kind of help with the flow of the document. But you're basically just walking through each of those study reports about how they contribute to the big picture and tying them all together. So I'd say start on the clinical trial protocol early. Think about how you're doing everything in the patients, and then go back and make sure that you're modeling all those things preclinically to whatever degree you can. And that's why I'm saying to start working on the clinical protocol early, because it really makes you think a lot more. Anyway, and I'll say divide and conquer. Ideally you have a project manager that's looking at the whole IND, and they're just saying, all right, here's all the different parts. This part is owned by this person, this part is owned by that person, and different people are responsible for different areas of content, and they're just working on different parts of the document at the same time. But it's also really important to have a lead decision maker. You can have a lot of cooks in the kitchen, but I've definitely seen cases where things really get bogged down, where everybody's offering comments, everybody's kind of providing very helpful feedback about what should happen or what content should be in there, but nobody's actually making the decisions. And nobody's moving the document to a final state. And that's what you really want to do, is not sort of this perpetual cycle of discussing stuff, but like, move it towards a final product. As far as the preclinical studies right now that everybody is right in the middle of and everybody's doing, I'd say think about all of this, about how that information is going to go into the IND. And you want to be very transparent with the FDA. You can't cherry pick and share with them the good data and not the bad data. Basically, if you tested your drug in some system, you need to share that data, good or bad. If you have gaps, studies are complete, or you get odd results, this can be okay. Whenever I'm, a lot of times I might have, 6 preclinical study reports that kind of go into the IND, but I'll say, look, these are the 2 that are really pivotal. Like one is determining our efficacious dose. One of them is determining our safe dose. Those are really the pivotal studies. Those ones have to be really thorough. They have to be really, hammered out and complete. You could have other studies that are kind of contributing additional information. But you can, say there are gaps, or the studies aren't necessarily complete, but they don't really need to be. You're just being transparent with the FDA. But making it clear to them, like, these are the studies that are really pivotal. I think it's important, any of your, especially your pivotal studies that determine your safe and effective dose, those ones you really need to archive all the samples that come out of those studies. And a lot of times, we can design a study and we'll say, look, we're only going to analyze these few tissues, but we're going to archive a larger set of tissues just in case a question comes up. And that, that's usually something that'll make the FDA happy. Same thing, you need to archive and retain all the study data, all the raw data, all the study records. And so you need to have a plan for this retention, and somebody has to have responsibility for that. One thing I can't emphasize enough is save a portion of any batch of your drug that you make, especially if it's being used for pivotal studies. If you can go back and do kind of analytical comparability of different batches, then a lot of times, that helps you mesh together, that make sure that all of your doses are calculated properly and everything. And if you don't have that, you're kind of at risk of getting asked to redo a study. And the FDA, best practice, the FDA would recommend you having some sort of a quality officer to oversee any studies. In my experience, with our INDs, anything that's outside of like a GLP study that is going to have it's going to be running under a quality umbrella. Outside of those, it's usually fine for the PI to take responsibility and sign off on studies. All right, last slide. I'd say ways that you can save money, which is important for all of us. We have limited budgets. Is if you have any sort of custom analysis that you need to do, some kind of custom assay, if you try to have a CRO do that, like a contracted lab, it's gonna be incredibly expensive. Anything that's not off the shelf for them, you're gonna get charged a lot. And so I'd say consider doing these in your lab. And even, for example, like a qPCR biodistribution study is, I've done a GLP toxicology study that's contracted before, and then I got a quote for over $1 million to do a qPCR biodistribution analysis. I could do it in my lab for like $100,000, and I could meet everything that the FDA actually needed. So my lab isn't going to be doing it under GLP, but so what you do there is, like, the CRO will say, okay, this is a GLP study. Here's a list of different things that are exceptions that are contributing to that report, but aren't done under GLP. And that can be okay. So I'm just saying things like drug potency assays, again, qPCR, biodistribution, expression analysis, immunology studies, device compatibility studies, things like that. The last thing, manufacturing, probably a lot of you recognize this. This can get very, very expensive and probably be the biggest expense of trying to get your clinical trial started. One thing to recognize is that the bar is lower for a drug being used in a first-in-human phase 1 trial. And a lot of times you might go to a contract manufacturer and they'll propose a lot of activities that are really suitable for a commercial product or a phase 3 product. And you can really scale back from that substantially, and reduce the expense, and also reduce your timelines. But you just have to know to kind of push back on that. So anyway, I'm going to end there so that we have plenty of time to go into our discussion and questions. And I guess I'll pass off to Ben to lead that.
Great, Steve. Thank you. That was a wonderful overview. Maybe I'll start just with a couple questions from some people who sent things in by email earlier, pretty related to your topic. You talked about it a little bit. I'm wondering, for the past IND submissions, who actually wrote all the sections? So you mentioned, having the CDMO contributes a lot of the data that goes into the CMC section, and you have a regulatory consultant who helps summarize that. What about all the other sections? How much of that was on you directly? How much help did you get from other people in various sections?
Yeah, so for us, again, going back to the preclinical study reports, if we're starting there, well, we have it divided up because I've got my clinical collaborators and, they're working on the clinical trial protocol. I'm providing some input, and there are some sections that have to kind of mesh with the preclinical work. But looking at module 2, in all the preclinical sections, again, it starts with the study reports or even the publications, the manuscripts. So if those are being done by people in my lab, then it's usually that postdoc or even that graduate student or that scientist they're writing the draft of that report, just like probably how your lab is preparing a manuscript. They're writing the initial draft, and then I'm the PI, so I'm reviewing that and editing that, and then we get it to a final state. And then we have our regulatory scientist that would give it one final read through and just make sure that nothing's missing. And then we'll sign off on it. And then you have each of those study reports. And in my case, when you're incorporating all those into module 2, like the preclinical summaries, it's kind of a mix. There's, in our case, our regulatory scientist, where I'll work with our regulatory scientist, and I do a lot that's hands-on of, kind of organizing the flow of the document and what are the points that we need to make. And so once we have that detailed outline, then it's a mix. Like the regulatory scientist or project manager or something can initially populate that by just taking the conclusion sections from the study reports and just populating that module. And then usually it's like me would go through and just, again, sort of edit and wordsmith the flow of that document. But it's a lot of work, and there's no way around that.
Takes a team to eat an elephant, I guess.
Yeah. And so you really need to give yourself months to get this together and start working on any sections that you can. Start working on those as early as you can, so it's not, you've got the work spread out over a long time.
Yeah, I wanted to follow up, I guess. I mean, Karen, if you want to jump in and add anything, let me know too. Okay. So another follow-up question on a similar topic, well, on a slightly different topic. You mentioned, being fully transparent with the FDA. When do, in terms of all the studies you've done with your drug product, when does that process really start? So let's say you worked on AV gene therapies. You've worked primarily with AV9, but let's say you were like our group, engineering capsids. Do you disclose everything to the FDA that you've done with the lead capsid and or the lead cargo if you haven't actually combined them together into your drug product? Or either. So at what stage is it that you start disclosing everything that you've done?
Well, here's the way I think about it is, the Interact meetings and the pre-IND meetings are there to help you. And so if you have questions, if you're not sure if something is okay, you've got some data that you might be concerned about. Generally, if I have a study that's inconclusive and we're still working on it, I'll just leave that out. I'll wait until we have an understanding of it before I present it to the FDA. But in the Interact meeting, if there's questions or concerns that I have, just go ahead and put them out in the open. And this is your chance to kind of check with the FDA, tell them this is what I'm thinking. These are potential concerns. These are the reasons why we think they're potential concerns, but we don't think they're big concerns, for example. And then see what they say, because what you don't want to do is have it where you're kind of holding something back that you think might be a concern and you don't find out until you submit the IND. That's the worst time to try to address a concern.
Yeah. But I mean, what if it's not about a specific concern? But for example, Sonia asked in the chat today, any study you do with your drug, share with the FDA. Does this also apply to proxy molecules? So how close do you have to be to the final drug candidate in order to enter the full transparency zone?
So
For
I
those
guess
of us who are doing a fair amount of iteration on our drug, what will ultimately be our drug product.
I'd say make a judgment call of, you need to, your final molecule, your final drug, like that's what the FDA needs to review. If you've got prior iterations or things like that, I wouldn't bring them up necessarily unless it's just that information is important for the FDA to understand, your current molecule. And for example, you might have like a previous version that you did a lot of work with. And we're doing this right now where we have some things where, we've been working with AAV9 and we're proposing to switch to a slightly modified capsid, right? And so we've got all of our data with AAV9, which isn't our final molecule. And then we're so we're still providing all of that data on the original, the prior molecule. And then we have a smaller data set on, sort of bridging to the new one. Because the final molecule, we basically don't have enough data by itself to really support everything that we need. So we need to include all the data from the prior molecule.
Great. Yeah, let's see. So I guess one other question from Eric Minical was on the email was, when GLP studies don't go exactly according to plan, if not all tox animals were dosed correctly, how do you handle this? Call attention to it or not? And at what point does it merit a conversation with the FDA? Or is there a disadvantage of submitting the IND as is and having the conversation after the fact?
So I actually have a concrete example of this. For my very first IND, we had a GLP tox study that was done in mice. And when we looked at all the biodistribution data, it didn't look like they did the injections right. And a lot of the animals effectively got underdosed. And so we reached out to the FDA immediately and scheduled a meeting with them, and we talked through it. And we had a smaller study in another species that we felt like had been done right. And basically we just talked through the risks and the considerations, and eventually we agreed on a path forward. But a lot of times, yeah. And I mean, I just had a recent study where like we did the biodistribution analysis and we did like a AAV9 antibody analysis, and we found that one animal looked like it just wasn't dosed. And that shouldn't happen, but it happens. And so I think whenever you have something like that, you basically just have a risk assessment. You say, this is what happened. This is what we're seeing. This is what we feel like the impact. And the impact is either minimal or non-existent, or else it has some impact and you're trying to weigh the importance of that.
So then if you think the risk assessment suggests that there might be a chance the FDA would want the study repeated, I assume that's a kind of a trigger to go talk to them first.
Yeah.
So let me see. Another question a little bit on a different topic, but you've talked about getting to filing the IND and writing that up. What about the relationship with the IRBs? And Sonia asked a question around, can you speak to the timeline/circular dependency of getting an IND and IRB approved? And are there bureaucratic or administrative steps that should be initiated long before you're ready to submit your IND?
Yes. I think every institution is different in terms of how they handle it, and Karen can probably talk about, how they handle it there. But, for us, we can we can kind of notify our IRB that something is coming. And we can notify our IBC that something is coming, but they typically will not look at it until we have the IND approved. So, and this does get circular because aspects of your study can change as it goes through FDA review. Aspects of your study can also change as it goes through IRB review. And so, my experience is once it goes through FDA review and the FDA is signed off on it, then the IRB is probably more likely to, I guess, defer to the FDA and kind of accept that it's okay for the FDA, but they may very well have changes that they need to make, especially in the informed consent. And so what you do is you have the IND approval, IBC approval falls somewhere in there. Then it goes to the IRB and you have IRB approval. And if there's any changes to the IND or consent or anything that the IRB stipulates, then you've got to go back in and submit an amended IND to the FDA. You just have to amend it with those changes. And I think that those are typically like non-hold kind of things. So, you just have to basically submit that and notify the FDA, but you don't have to wait for them to review it. If it's something substantial that you think, then it's probably best to wait or give the FDA a certain amount of time before you initiate your study.
Right. Thank you. Another question from before today. This is on a different topic. What potential streamlined regulatory paths for editing therapies, like different guide RNAs for the same disease or different diseases, exist now, and what may change under the new platform technology designation? Maybe this was a good one for Kiran.
Yeah, maybe I'll put it to Kiran first.
Yeah, I mean, and the short answer is there is nothing yet, right? So there's been a lot of talk. The FDA is increasingly showing some openness to the idea of streamlining, but I'm not aware of a single example where this has actually happened. And it won't happen until someone goes to the FDA with a specific proposal to do the streamlining. And, there have been meetings recently involving people from academia, involving people from industry and so forth, around the concept of platform gene editing. And the strong sense I get in talking to folks, from the companies is that none of them are all that excited to be the ones to really push this forward. If anything, I get the sense that they are relying on us in academia, in Somatic Cell Genome Editing Consortium or otherwise, to be the tip of the spear and really be the ones pushing this with the FDA. So the answer is it depends on us. We're the ones who are going to have to very likely go to the FDA with specific proposals. Hey, we have these two very similar drug candidates. They only differ by the guide RNA or XYZ. Will you allow us to streamline? And then it'll be up to the FDA to decide whether that's acceptable or not. And then, if they don't like the first version of what you put before them, then you got to come back to them again and give, a different variation. And then we're just going to have to feel our way through this. And that's one advantage of this group, the Somatic Cell Genome Editing Consortium, that we can do this in a more coordinated way than any one academic group could do it alone, right? We talk to each other. We have meetings like this. And, we'll learn as we go, and the FDA will learn as they go. We're starting to make incremental progress, I sense, with some of our Interact meetings from this very, consortium. But the shorter answer is, there's no guidebook, there's no guidance, draft guidance or final guidance or anything whatsoever that's been released by the FDA. Maybe that will come in time. Now, with respect to the guidance, I guess it's still a draft guidance. On platform technologies, something that's very important to recognize in reading that carefully is that the platform designation will only apply once there's actually a clinically approved product that's being marketed, right? So, and it's very clear about that. Only when that happens will the FDA consider a platform designation for other drug products, essentially from the same manufacturer, using essentially the identical processes. And as that actual approved drug that is on the market. It doesn't mean that there is an opportunity to streamline for us in the gene editing community, but you do have to make that distinction. What we're proposing to do, what we're pushing on, is not formally discussed or included or envisioned in the actual platform designation as the FDA has proposed it, because none of us are in a position where we have an approved drug. In fact, nobody is in a position where they have an approved drug, at least an in vivo drug, that editing drug, that would permit the platform designation. Maybe on, the AAV gene replacement side, there are enough companies now that have done this and have approved drugs where they could take advantage of that. Is there some way us in academia could ride on their coattails and take advantage of that? Maybe in theory, but knowing the complexities of how industry does things and confidentiality and all the considerations that go with that, I would think it challenging for an academic group to somehow be able to attach itself and fall, be able to fall under that umbrella of a platform designation that has already, under the auspices of what a company actually has approved. Again, it's not IND clearance. It's not first in human clinical trials, early phase clinical trials. It is, at least the way the draft guidance reads, is an actual approved product. So that typically will mean in a commercial entity.
But I guess outside of that though, in what, maybe if it's not like the formal designation of a platform technology from the FDA standpoint, I think the concept of that is something that we can leverage. And I think that the FDA is likely to be receptive to, if we can cite each other, or cross-reference each other's INDs, or if we can publish or make documents public and then reference them, or even take confidential, unpublished information that somebody's willing to share, and you can include it in your IND. We've done all of those things, and it's where you can leverage data from other studies, I think, to cut back on what you're doing for your product. And we, say for AAV biodistribution data, I mean, we do that all the time, where we just cite that there's all this published literature out there, and we don't need to do this extensive biodistribution study, and the FDA has really always found that acceptable.
Yeah, fully agree. I mean, there's plenty of room for us to maneuver. Don't get me wrong. It's just it won't be under that formal, like guidance of a platform designation. It'd be something a little bit different. A lot of the same principles could very well apply, but we would be doing something a bit different. Yeah,
and in general, because a lot of what we're doing is, I'd say, cutting edge. Then if we kind of sit back and wait for the FDA to tell us what's okay, then we're going to be waiting a long time. We collectively are the experts. And so, I think we need to be telling the FDA what we think is acceptable and providing, really clear rationale for that, and have it backed by data. And, and they're always going to have other perspectives, and they're going to have access to data that we don't. But, figure out what makes sense for us first, and then present it to the FDA instead of the other way around.
Steve, I think you've developed, quite a playbook, I think, through your extensive use of AV9 and doing gene replacement therapies, and ultimately, even your recent SPG 50 N of 1 therapy that you miraculously did in under 3 years, from diagnosing, or from the patient that was diagnosed to actually dosing them. So how much of that do you think you will be able to leverage for your, a gene editing therapy? I mean, how different is it going to be? Where is your interaction with the FDA deviating? Obviously, you're going to have to do off-target analysis and other things, but like, how is that playbook transfer? And do you think you're going to be able to go with a similar speed with some of these newer therapies?
I don't. So when we move into gene editing, I don't think we're going to move at the same speed. But I think that we leverage the general toxicities around the vector, which we've got, like a decent amount of information on, and one more small non-human primate study is really not going to improve our understanding of that. So the safety around the vector itself is something we can leverage, the biodistribution of the vector itself, manufacturing, we can leverage all of that. So that really, you start to strip away a lot of the different factors and concerns, and then what you're left with is really, what are the immune responses or safety considerations of the editor itself and the off-targeting effects. And then you just address each one of those on its own. That's really our strategy.
Does that mean that you are going to try to minimize non-human primate studies to get that information? And if so, are you confident the FDA is going to allow that type of approach with these new cargos?
Yeah, well, I think in our Interact meeting, we kind of made that exact argument that doing an off-target editing study in non-human primates probably wasn't that productive. And that assessing vector biodistribution wasn't going to be that informative, or safety around the AAV. But so we did propose a small non-human primate study, but it was mostly because it was a dual vector, and we hadn't ever done that before. So we did want to look at the expression of the dual vector and see how that translated from mice. But it was mostly around the immune responses. That was the safety component of it, and that was really the rationale.
So how I did want to ask about that as well. So in both of your experiences, but maybe mostly you, Steve, because you're delivering something that's going to be expressed permanently. How has the FDA viewed the importance of various types of data one might collect around immunogenicity, given the challenges of looking at this in non-human species?
I just, based on experience, I don't really place a lot of value on immunology concerns in a rodent study. I just don't think the rodents are that informative. My opinion. I think that we've modeled expressing foreign genes in non-human primates. So we probably could have leveraged that. I mean, we might even have been able to try to push them into not even doing a non-human primate study, but it started to fall into, what do I think I might be able to get away with, and what am I actually comfortable with? And I felt a level of comfort of putting this into non-human primates because it was different enough than anything that I'd had experience with before.
Yeah, and with respect to what I have experience with, which is primarily messenger RNA, lipid nanoparticles, the same immune issues, as you pointed out, Ben, are not there. If anything, there's more emphasis on acute reactions, like innate immunity to the lipid nanoparticle itself, and looking at cytokines and complement and things of that sort. That's where, at least in my experience, more of the emphasis has been, rather than long-term, cytotoxic T lymphocyte responses or things of that sort. I will say that context is important. So, as many of you know, we are trying to undertake a sort of compassionate use, individual patient expanded access, IND. And in that context, the FDA actually showed quite a bit of flexibility. They did not insist on a non-human primate study. We're doing one anyway, but they actually said you don't necessarily need to do a non-human primate study. Rodents would be sufficient for this context. We're talking about a very severe condition, a very high level of unmet need, very limited life expectancy, that sort of context. So that context does become important in thinking about, what is the FDA is going to require and what they'll give you a pass on.
Yeah, and that's something important even when you get into your toxicology studies and when you start getting results, because for example, like the SPG50, the one that moved really fast, we did have toxicity findings in non-human primates and rats, so that we had toxicity that was graded adverse. didn't kill the animals. They really didn't have much in the way of functional issues, but there was toxicity like around dorsal root ganglia. And so we did the risk assessment and more or less said, yeah, there's no treatment available for this disease. It's very devastating. And the risks of the treatment, I think, were outweighed by the need and the potential benefit.
Great. I think I'm gonna, unless you guys have anything else to add right now, I'll switch and start going through some of the online questions, and then maybe we can even open it up to people asking questions live if that's possible. I don't know if it is, Mindy. All right, so we have a question from Yusuf. What are the phase 1 fit for purpose exceptions you were able to make to get lower quotes from GLP and GMP CROs?
Wow. This kind of gets in the weeds a bit, but for example, if you go to a contract manufacturer, then consider it from their perspective. What happens if a batch fails? Then it gets really awkward, right? And it has to be repeated, and who has to cover the cost of that? And so they're going to want to mitigate their risk as much as possible by, fully developing, qualifying every analytical assay, and doing a lot of engineering runs to build up manufacturing history, and then saving a lot of retains for each batch, and so that mitigates their risk. It increases your expenses and your timeline substantially, but it mitigates their risk. And so you really have to push back on that. And sometimes that means basically saying that you're willing to take on a certain amount of risk. And that risk might be a difference of a 1% chance of it failing versus the cost doubling, but a lot of times, like fit for purpose, when you go down the analytical testing of your product, like I'm going to go use AAV. So the vector genome titering, like that has to be locked down. That has to be rock solid. Things like the percent full, and the potency, those should be pretty solid. But then you get into a lot of the different impurities that are being measured. Again, some of it's so they might have an off-the-shelf assay that they can run on your product, but then they want to fully kind of qualify the assay, this off-the-shelf assay using your product, and that's probably not necessary. You can accept a very minuscule risk that something a little bit odd is going to be happening with your product. And also whenever they're doing I've definitely run into this where we talk to them about the vial disposition. So they make your drug product and they vial it, and what happens to all those vials. And then you start to have, okay, here's all the ones that are used for analytical testing. Here's the ones we're setting aside for stability testing. Here's the ones we're setting aside in case you need to repeat the stability testing. And here's all of the retains. And then they do all that. And then at the end, you don't have any drugs available to dose your patient. I'm honest to God, I've had that happen plenty of times. And then you have to go back and say, all right, we're willing to accept a certain amount of risk that we don't have. Here's like a few vials of retains that are available across all of our stability studies. And, here's it. Anyway, there's certain regulatory requirements you have to meet, but especially if it's being used for a phase 1 material, again, that bar is a lot lower. And this is where, it helps to have a good CMC consultant that can help tell you, what do you really need versus what do you want?
And are there similar opportunities for the GLP studies with the CRO?
Yeah, and again, it was for us, we always leave the histopath to the CRO, all the animal work to the CRO, but when it gets into sample analysis, we pulled the FDA guidelines in terms of doing the biodistribution analysis. We could develop and qualify the assay to meet those requirements, and we can run it ourselves for, again, literally like a tenth the cost. So I'd say all the in life things that have to happen with the animals, like all that needs to happen with the CRO. But when it gets into the sample analysis, that's when you can kind of step in.
Okay, so Chris Saha has a question. In the IND, do you describe and anticipate protocol amendments during the first in human trial? If so, in what sections? Module 5. We are considering guide RNAs from those used in our pivotal studies and expanding the patient pool via protocol amendment.
Yeah. I'll weigh in. Then I'd like to hear what Kiran has to say. My thought would be, try to go in with an IND that is basically as simple as you can, and just focus on getting the initial approval. And you can have a section in there where you that's a little bit forward looking, where you can tell the FDA, once this IND is active, these are some of our this is how we envision the long-term development of the product. I mean, you should have that in there. So I would say to, focus the IND on something relatively small that that you're going to do initially. Have that section describing the possible long-term plans, and we do this. It's not a big deal to amend your IND to increase the number of patients, or at least for us, it wasn't necessarily a big deal. So if you're talking about expanding it to include other guide RNAs, again, my advice would be get your approval on your one product, and then you can have a separate discussion with the FDA on expanding amending it and expanding it. I don't know what you think, Kiran.
So I'm chuckling because our plan is actually to do exactly the opposite. To go into our pre-IND meeting and propose just, from the get-go, we want to do, a batch of 5 variants. And here's the plan for the full set, gold standard IND enabling studies for variant number 1. And then here are some, less rigorous studies, mostly in vitro studies for variants 2, 3, 4, and 5 using essentially the same editor. Will you accept this? And if they're going to put their money where their mouth is, and everything that, Peter Marks has been saying, and increasingly others at the FDA have been saying, then they'll work with us with that. If they really don't like it, then they will let us know, and then they'll make it clear, no, we want you to just, focus on the one variant first, and then we can talk about how, like, you could make subsequent amendments. And then, we'll go forward with the ID based on what the FDA seems, based on their, explicit verbal feedback and written feedback, what they will accept. I mean, you can push after the fact, or you can push from the get-go. And I don't know what the best strategy is, but we're going to try aggressive and see what happens. And we'll all learn from it one way or the other.
Yeah, that's great for you. Well, for you to be a trailblazer, great for us. Hopefully that goes well. All right. Let's see. Sonia had a question. Steve, when you say to start early, does this mean initiating an application online in an online system long before you're ready to submit, like as early as now? I think we don't hear you.
Yeah, not starting something with the FDA in terms of outside of like your Interact or pre-IND meetings. I'm just saying you could have an internal document that your group is working on, where you're all, you're working amongst yourselves to fill in different components and start to populate things. because ultimately you're going to develop that whole document internally. And I should say, once you finish populating everything in the IND, I mean, it would take a regulatory scientist probably a few weeks to kind of QC the document, make sure everything's linked together and everything. So, because the INDs are usually thousands of pages.
Okay, there's another question. This is from Megan, are there any additional considerations for interactions with the FDA that you anticipate in the future? I think this is probably a good one for both of you.
I mean, I think we need to keep doing what we're doing in terms of, there's the regulatory work group that we have set up with the SCGE, and to, as people are coming up on their regulatory meetings, as much as you're comfortable, share your questions with the group ahead of time. Like, let's talk through them so we can learn from each other and pool that knowledge and, ask things that benefit, cross-cutting across our projects.
Yeah, I would essentially, say the same thing. I mean, the formal interactions, they're the interact meetings and the pre-IND meetings. Many of us have had interact meetings, and in the fullness of time, I expect most, if not all of us, will have pre-IND meetings and be able to get formal written feedback and verbal feedback that way. There are, of course, less formal interactions, sort of the back channeling that happens at, in-person SG meetings or in other, other forums. Forums like the one that was put together by the Alliance for Regenerative Medicine, ISCT, a few weeks ago that I think some of us were involved with. And so that's another good source of information. And then, as many of you, I think, have heard, those of you at least who participate with the regulatory work group, we now actually, by we, I mean the NINDS, has a memorandum of understanding with the FDA. And so that just became active, as I understand it, and that will be a channel where we, via that MOU, can ask questions of the FDA and get answers, hopefully, to those questions.
Yeah, one thing I'll add is, keep in mind, the FDA kind of has their set formal forums for interaction, and before the IND, we have the interact meeting, we have the pre-IND, especially following up on a pre-IND where additional questions have come up, or we've gotten some weird data, or we're having a question with our contract manufacturer about how many vials we need to retain, or, things like that. We've just sent in an information request that references our pre-IND, and the FDA is not obligated to respond to that. But for me, in every single instance, they have. They've always been incredibly helpful, and usually they've agreed with us. So don't feel like you have to hold back. I would not advise pestering them about every little thing. But if you really have a decision point that you need to make, then just send them and it can be something it could be like a little 1-page write-up, or something where you're asking them a very specific question.
There's a couple questions from Zheng. In your preclinical study, how many people do you have and what roles do they play? So I guess this is asking, like, in your group, collaborators, clinical collaborators, how have you moved this forward?
I mean, we've got an IND that we're getting ready to submit this week, that we've been working on for a while. And it's really, I mean, the core team in my lab it's like 3 people on the clinical side working on the protocol with me. And then it's me, a project manager, a scientist, and it's like maybe 4 or 5 people on the preclinical side that are contributing information. So, I mean, it's not this huge effort. And then, like I said, we have an external CMC consultant that we bring in that, that we've been working with for years. So it doesn't necessarily, you don't need this ginormous team. But I think it's important to be organized and kind of stage the information so it's not everybody dropping everything and not working on anything else for 2 months.
The follow-up to that, I mean, how have you been able to keep the same, largely the same group of people working on many of the INDs that you've worked on, or is it really project specific?
It's usually different people on the clinical side that are more disease focused. And on the preclinical side, like I said, it could be the postdoc or the research scientist that's really leading the project in the lab. Like they're the one who's, yeah. So I guess I'm the common glue. My regulatory scientist and my project manager are the common glue.
Got it. So he also asks, where did you do your NHP studies? If you're say that.
Yeah, well, we've worked with Charles River. We've worked with a company called MPI that got bought out. We've worked with a company called Northern Biomedical Research. I don't know. I mean, if you want to reach out to me offline, I've had experience with a few different CROs, but I would recommend putting together an outline of the study of what you want to do and going out to multiple CROs and get quotes. Yeah.
Okay. Great. Sonia asked another question. Have you ever been put on partial hold but been allowed to proceed with part of your clinical plan? Is there a downside to proposing to dose a wider group of patients knowing FDA may come back and say, no, you can only dose the most severe patients? I guess those were two questions, but do you want me to?
Well, I know exactly where she's coming. I know exactly where she's coming
Yeah.
from on this. Yeah. Yes. Sonia, I'll just say, so we had a pre-IND meeting recently for a form of Charcot-Marie-Tooth disease that is not fatal. It's debilitating, but it's not fatal. And we had preclinical data that showed that early intervention was much more effective than treating later in the disease course. And so we just discussed that in the pre-IND. We outlined a plan of, I think in this case, we said that we would dose 2 adults. And then we would open up the enrollment to basically anybody, I don't know, it might have been like older than 3 years old. And they agreed with the plan. So I think if you've got data that the treatment will be much more effective when treated earlier, and I can't speak for the FDA, but I think that they will understand that. And so just outline your plan, and especially if you're going into consenting adults, I think that gives you a lot more leeway. So
You,
I mean,
you said you asked some of these kind of questions. Sorry to interrupt.
Yeah,
At the pre-IND stage, have you ever asked questions like that at the interact stage?
I've had very few interact meetings.
Right.
So, but I think that it would be appropriate to ask it in an interact meeting.
Yeah. Maybe I'll ask another question. We have a little bit more time. What specific feedback did the FDA provide during the interact or pre-IND meetings that substantially altered your initial approach to your IND? Do either of you have examples of that?
Yeah, I can give you one example. This is with the SCGE funded program on PKU. We had proposed doing a definitive IND enabling study, tox study with the disease model. This is a PKU model. And we asked whether that was acceptable. And what we were told in the interact meeting was, we'd really prefer you to do it in a wild type animal model. So that's a pretty substantial change. And so, my experience with this, not only with respect to the SCGE funded projects, but also exposed to this on the industry side, is they can ask for very substantial changes, particularly at the pre-IND. Meaning the interact meeting, the FDA tends to be dilatory in the sense that you ask a lot of questions and then they will often just say, well, that's something that we can discuss at the pre-IND meeting. It's hard to get a lot of concrete stuff in the interact meeting. In my experience, the pre-IND meeting is where the rubber really hits the road and you propose things and they will definitely come back. and say, we don't like the way you've designed this, please do this instead. That could be with the off-target analysis. That could be with, the preclinical studies, whether it's, the farm tox studies or whatnot. It could be with the clinical trial design. It could be really any aspect of it. And they'll make it very clear what they think, and they'll give you their written responses, and they'll expect you to respond to their written responses before they would let an IND application go forward.
What was their rationale for wanting you to do it in wild type animals versus a disease model?
Yeah, the rationale was that, which I didn't fully understand, was that the disease model might not give an authentic assessment of the biodistribution and the toxicology of the drug product, which didn't totally make sense to me because, well, if we're treating a patient with this disease, isn't that what you really want to know? But at least the reviewer we had in that meeting, that is what he told us. And to be honest, I'm not displeased, because if you're really thinking about a more platform approach where you want to cross across diseases, wow, it's convenient to be able to do that tox study with the basic LNP in a wild type animal and have it apply to all the diseases if they're really going to let us do that, rather than having to do it over and over again in each disease model.
Yeah.
Yeah, I mean, I'd say 90% of the time, whenever they raise concerns or comments, I understand where they're coming from, and I can, there's been a couple of times when, I mean, I don't know, I think one time we were doing something like a pretty low dose injection of AAV into a nerve. We were proposing to do that, like a direct injection, and they wanted us to do a complement analysis in our tox study and look at DRG toxicity, and we were nowhere near the DRG, and so none of that made sense. And eventually we just, usually I can argue things and get it straightened out, but that was like a case where I don't know, they just wouldn't budge. But I think probably the one was we were going into a disease model that didn't have any disease symptoms. It was much, much more mild than the human disease. There really wasn't a good animal model, and we really got hung up because it was a proposed pediatric trial, and we had to show the prospect of a kind of clinical benefit in the animal model, and no animal model existed to be able to do that. So we were focused on a biochemical readout in the animal model. They kind of pushed back and said, well, what links that biochemical marker to a predicted benefit in the patient? And so that actually prompted us to do a clinical study where we actually looked at a spectrum of different severity of patients and looked at the levels of this biomarker and generated some data that reinforced that improving that biochemical readout would predict a benefit. So, it felt like a little bit of a extra hoop to jump through, but I understood where they were coming from. And I guess there's common things like, we usually propose not to test in non-human primates, and for all of our recent interactions, and I'd say 90% of the time the FDA agrees with us. But we have had a situation like we were working with a neurotransmitter, and then they wanted us to do a non-human primate study, just considering that a primate, might be more complex than a rodent there.
That makes sense. So the farther outside the box you are in terms of just expressing a missing gene, I suppose.
Yeah. Well, and I guess the other thing is, if you're moving out of rodents, also don't assume that you need to do non-human primates.
Yeah, sure.
And we had something where, due to anatomical reasons, like the monkeys were not, it was actually better to do the study in a dog. And that's what we ended up proposing, and that was fine. We needed to do it in a large animal model for kind of anatomical modeling, but the monkey really wasn't the best model.
Do either of you have, or both of you have, things that you've learned that have really allow you to just save a lot of time in the future? Steve, I know you've gone super fast with one of these. What words of wisdom could you give the consortium on really staging all of the efforts that we're trying to do? I mean, these are very complicated, right? And I think what we're doing in this program is even much more complicated than a gene addition, but that's already challenging. So do you think the roadmap? I know you published your nice paper with the spasticity gene therapy and had a nice timeline of all of the things that you did in the events. How well do you think that maps things out for this group, or are there just a bunch of additional things that need to be done in parallel?
I think that actually maps things out pretty well. As long, you're using a different capsid, so that's a different variable, right?
Different capsid, completely untested cargo. So
yeah. But I mean, I think it can follow a certain track. Except you've got to address all the off-target editing issues. And so I think the consideration in terms of speed is how much confidence do you have, I guess, in how the technology is working, that you're comfortable just doing that in parallel with the other activities versus sequential. Once you get into sequential, then everything just takes a really long time. So if you feel like you're checking a box, then do it in parallel. But if you feel like it's really gating,
yeah.
then do it sequential.
In terms of off targets, I mean, this is a question for both of you. What do you think the off-target analysis? What has the FDA? What have they really considered to be the best assays or the best ways of looking at this, or is it just really so bespoke for every single different type of editor? And also just like cell types, and if a lot of this is going to be done in human cells, I mean, if you know the biodistribution of your delivery agent, how many different cell types are you running these assays in, depending on where you think your product's going to go?
Yeah, so it's evolved a lot over the last few years. I think a few years ago they were very, very rigorous and almost to ridiculous standards, and making companies, spend literally millions of dollars. In one case, putting a program on clinical hold for 10 months just to get more off-target data that did not actually change the profile of the drug product whatsoever. It's a huge waste of time and expenses. And I sense that, Peter Marks and the agency recognize that they were holding way too high a bar and have somewhat relaxed in their approach to off-target editing. There's no one size fits all, of course, but they do have some preferences. They want to see orthogonal assays. They don't want to see, all your off-target profile be pinned on exactly one assay, because there's no perfect assay. I think we all appreciate that. So they like to see complementarity, two orthogonal methods that together give you a more comprehensive view of what off-target editing profile looks like. With respect to cell types, they still very much, as they were a few years ago, they're still very much intent on tying the off-target assessment to biodistribution. And so you do your biodistribution, whether it's rodents or non-human primates or whatnot, and then you use that data to nominate which cell types there does appear to be delivery of your editor, messenger RNA or whatnot. And then those are the cell types in which the FDA will typically expect you to do off-target analysis. You can generate the list without any reference to cells, right? So some of this depends on how cleverly you set it up, right? If you prefer to use techniques like guide-seq or whatnot that are very much cell dependent, then you might find yourself in a position where you're doing that assay, that discovery assay or nomination assay, over and over again in different cell types. Whereas if you use methodologies that are not tied to any particular cell type, but are genomic DNA or whatnot that you just isolate from cells, and so you're generating your nomination list in a cell type agnostic way, that can be advantageous, because then you just take your list and then you do your ramp seq or your hybrid capture or whatever your high throughput method of going through all the off targets. And here I'm talking more about nucleases and base and prime rather than epigenome editing, which is kind of its own thing. And then it becomes much easier to deploy that across the different cell types and then generate that data. Yeah, so I think those are the general guidelines. And in terms of specifics, you can always look to the FDA's guidance that was published early last year, or was it early this year? I don't know, I lose track. Maybe it was early last year. The final guidance on gene editing, genetic medicines. I can't remember the exact name of it, but it's there, and it goes through in pretty detail what kind of things they're looking for from the off-target analysis.
Great, thank you. So there's another question on a similar topic. Have you ever done AV integration studies for both target organ and other organs? Steve, this is probably to you.
Not necessarily. I mean, we've definitely done biodistribution studies, but not. No, we haven't ever had a reason to do an extensive integration study.
Sonia also asks, it seems like a disadvantage of moving away from NHPs for large animal studies is that so many fewer, oops, moving on me. So many people have the animals and know how to do the procedures. So did you run into challenges when you moved into the dog, and maybe you can speak to how that process went for you?
Yeah. So this is an important point, because sometimes people are doing kind of exotic injections. I mean, and really like a intrathecal injection can be exotic depending on who you're working with. So I would, whenever you're talking to different CROs, then, ask them flat out, like, what is your experience? Like, how many studies have you done where, you've injected via this route? And, in our pre-IND meetings, the FDA has asked us, and this is good. Like, they've asked us for, if we're doing like an intrathecal injection, which is also difficult to do in mice, is they'll call out, give us documentation that the technicians doing the injections are actually proficient. And so that's actually useful when you go to the CRO, because you're like, hey, the FDA needs to see evidence that your technician is proficient, so what does that look like? I mean, we just had a study, and it was just like 2 non-human primates we were doing with an academic group, and we did the biodistribution analysis, and it was really clear one of them had a good injection and one of them didn't. And it was a group that felt like they knew what they were doing, and we felt like they knew what they were doing. And it's just, unfortunately, a lot of those injections, like, again, we do the lumbar intrathecal injections a lot. They're pretty routine in humans, but they're not routine in animals. And one of the ways that I've seen that it can be mitigated to some extent is to actually like implant a catheter and inject using an implanted catheter that can kind of have the placement verified much more thoroughly. And then you've got to, kind of discuss, risk assess that you're using a device that's different from what you're using in humans and, whatnot. But anyway, sorry, that's, the devil gets in the details. But I'd say, importantly, just when you're reaching out to your CROs, ask tough questions, ask them what their experience is, and don't let them kind of hand wave and talk around it.
Yeah, that seems like good advice. I think we're almost up on time. If there are no additional new questions, give everybody a couple minutes to ask anything else. But Steve and Karen, if you have any last words of wisdom, happy to give you a second. Otherwise, we can wrap things up.
Yeah, I would just say, I mean, I already sort of touched upon this, but the only way to truly know what to do is to engage with the FDA. And the more, formal official interactions you can have with them, that you can contrive to have, the better, because that's where the true communication and true expectations will be communicated to us.
Yeah, and I guess just in terms of the experimental strategy, what you're doing, how thorough you think things need to be, and what your risk tolerance is. I always say, what would I do if it was my child? What would I do if it was my brother or sister? And understanding the risks and the benefits, and just kind of go with that compass instead of just always saying, what does the FDA need? What does the FDA need? Start off with, what do you need?
Great. All right. Yeah, I was just reading Sona's comment. Yeah, I think I would second that a little bit. But maybe in the future we can have these with actual live questions from the audience. But I think this has overall gone really, really well, and I think I've learned a lot. I'm hoping the audience has too, and gotten a lot of their questions answered. So thank Steve and Karen for really a wonderful discussion, and the Inminion team for all the organization of this. Thank you so much.
Yeah, and Ben, thanks for stepping in to moderate. Appreciate
Yeah,
it.
no problem.
Yeah,
No
that
problem.
was
My pleasure.
Absolutely.
that was very helpful. Thanks, Ben, and thanks for Karen, who was helping to capture those responses. And thank you to Sonia for all the questions and for the feedback. So we'll talk about that internally, and how to do this better for everyone next time. All
Thank
right.
you, everybody.
Yeah, thanks everyone, and have a great rest of it's only Monday, so great rest of the week and happy holidays.
Take care.
Bye.